Another type of cell division that is required to make gametes or sex cells like sperm and egg cells is called meiosis. And meiosis is a slightly different process than what you see in mitosis. And it also involves an event called crossing over that can increase the genetic diversity of the offspring. So remember that in humans, our body cells have 23 pairs of chromosomes. Those chromosomes are called homologous because they have genes, the same genes for the same traits in the same order on the chromosomes. One member of each pair of your chromosomes came from each parent. One came from mom, one came from dad. In humans, the sex chromosomes X and Y are actually different in terms of their size and of their genetic composition, but the other 22 pairs, the autosomes, are the same size. They are definitely homologous to each other. Homologous chromosomes are the same length, they have the same position of their centromere, and locations of the genes are in the same place. A locus is the position of a gene on a chromosome. Sometimes you have different versions of the gene, a dominant or recessive version of the gene that can be found in the same locus, one from mom, one from dad. Okay, so this shows a pair of homologous chromosomes that these are actually duplicated chromosomes made of two sister chromatids, and it shows the loci of the different genes there in the same place on the pair of chromosomes. But one of these chromosomes came from dad and one came from mom. So in order to accomplish sexual reproduction, you have to make sure that you have the right number of chromosomes in cells. If you think for a minute what happened in mitosis, remember the chromosomes duplicated and then separated so that each daughter cell gets the same number of chromosomes. But what if you took two of those daughter cells and put them together? The daughter cell that would result from that would have twice the number of chromosomes, and that's not going to be the same species. So we want to make sure that the offspring has the right number of chromosomes. Generally speaking, organisms' life cycles has a sequence of stages leading from the adults to the adults of one generation to the adults of the next generation. A lot of animals and plants, including humans, have what we call diploid cells. So that's body cells that have two sets of chromosomes, one from each parent. That's what we're used to thinking about in terms of ourselves. Meiosis converts those diploid nuclei into haploid nuclei that have only one set of chromosomes. This is a specialized type of cell division that only occurs in your sex organs, producing gametes, the sperm and the egg cells. When we unite the sperm and egg cell together in fertilization, we produce a zygote that has the diploid chromosome number and therefore one set from each parent. So that's how you got one set of chromosomes, one of your pairs of chromosomes from each parent to begin with, is by means of fertilization. So this just shows the generalized life cycle, okay, showing that meiosis occurs in the ovary and the testis of the parents, okay, and that's going to produce egg cells and sperm cells that have the N number, the haploid number, 23 chromosomes. When they fertilize, they become a diploid zygote with 46 chromosomes that can then grow into the adult to start the next generation. We refer to haploid numbers of chromosomes as N, because that's just the basic number, and then diploid is 2N. That way we don't have to worry about how many chromosomes an organism has. We can still talk about whether it's haploid or diploid without worrying about the actual number of chromosomes if we refer to the chromosome number as N or 2N. Gametes have one set of chromosomes, okay? And so all sexual life cycles have an alternation between a diploid stage and a haploid stage. In humans in particular, the haploid stage is only the sperm and the egg cell. But producing haploid gametes prevents that chromosome number from doubling every generation. Because if you think about it for a minute, you have 46 chromosomes. If two 46 chromosome cells join together, that would be 92 chromosomes. And then the next generation would be 184, and it would just keep on getting bigger and bigger, and that gets kind of unwieldy after a time. So here's what's going to happen overall in this process. We've got a pair of homologous chromosomes, one from dad, one from mom, in the diploid cell. They're going to duplicate during meiosis. Okay, so you've got the sister chromatids here. And one thing that can happen is in meiosis you could separate out the two duplicated chromosomes into the first division into two separate cells, and then the second division separates the chromatids into separate chromosomes. So from meiosis you're going to get, rather than 2 daughter cells, you're going to get 4 daughter cells, but each daughter cell is only going to have half the number of chromosomes as the parent cell. So this is the type of cell division that produces haploid gametes in diploid organisms, and then the two gametes are going to join together to produce the diploid state in the zygote. There is a duplication of chromosomes in meiosis, just like happens in mitosis. However, in meiosis we get 2 consecutive cell divisions instead of just 1 cell division that we see in mitosis. So because there's 1 duplication of chromosomes and then 2 separate divisions, then each of the 4 daughter cells is going to have a haploid set of numbers of chromosomes rather than diploid. Now the stages of meiosis are very similar to the stages of mitosis. They involve prophase, metaphase, anaphase, and telophase, but since we have 2 divisions, we have to name them a little bit differently. So we talk about meiosis 1, the first division, and meiosis 2, the second division, and each division has prophase, a metaphase, anaphase, and a telophase. So in prophase 1, these are the events that occur in the nucleus, okay? The chromosomes coil up and become compact just like they do in mitosis. The homologous chromosomes come together. This is something different than what happens in mitosis, because in mitosis the homologous chromosomes don't pair up. The arrangement of the chromosomes is just random. But specifically in meiosis, those homologous chromosomes pair up together, and so they have the 2 doubled chromosomes that come together. This is a process called synapsis. And then you've got basically a group of chromosomes that has 4 chromatids. This is called a tetrad. And then during this phase, in prophase 1, there is an exchange of genetic material from non-sister chromatids by this process called crossing over. Here we have our interphase where the chromosomes duplicate. So far this looks just like mitosis, okay? But in meiosis 1, rather than just being individually separate from each other, the homologous chromosomes pair up together and form a tetrad, and then in that tetrad there is a crossing over between non-sister chromatids. So in other words, blue is going to cross with red and red with blue. Okay? So in metaphase 1, they're still attached as a tetrad, and they align up at the cell equator, just like in metaphase of mitosis. In anaphase 1, though, rather than the chromatids separating, the homologous pairs separate to each other. The chromatids of each homologous chromosome are still attached together, but the homologous pair separates and moves toward opposite poles of the cell. So here we have our tetrads lined up together. This time, instead of a spindle fiber from each pole attaching to from both poles attaching to a chromosome, they attach either to one or the other, and then those are separated or pulled apart in anaphase 1. In telophase 1, then those chromosomes have reached the poles. The nuclear envelope reforms around the chromosomes, and then you've got each nucleus has the haploid number of chromosomes, even though the chromosomes are still double chromosomes. They're still duplicated, made of 2 chromatids each. So here's our telophase 1 and cytokinesis that occurs just like similar to what we saw in mitosis. But this is going to lead now to the second division, or meiosis 2. Meiosis 2 is going to follow right on the heels of meiosis 1 without chromosome duplication. Each one of those products is going to enter meiosis 2, and the first phase, of course, is going to be prophase 2. So once again, the chromosomes are going to coil up and become compact. Sometimes they don't even uncoil. It just depends on the cell. The nuclear envelope, if it formed, is going to break up again. And so we'll end up with, here's our prophase 2, okay, nuclear envelope disappearing. The spindle is forming, and the spindle fibers start attaching to the centromeres. In metaphase 2, we're going to see this time there's a spindle fiber from both poles that attach to each chromosome. And then in anaphase 2, the chromatids are going to separate to become separate chromosomes, and then that's going to lead to haploid daughter cells from all 4. So here's a picture of 2 lily cells that are undergoing meiosis. This one's already at that stage, is already at anaphase, looks like, and then this one's or at telophase, and this is at anaphase here. And so very soon this will be 4 separate cells. So in metaphase 2, the duplicated chromosomes are on the cell equator, or the metaphase plate there. In anaphase 2, the chromatids will separate and move the chromosomes to opposite poles. And then in telophase 2, they're going to reach the poles, have a nuclear envelope around each set of chromosomes, and then you'll end up with 4 haploid daughter cells. Now, there are a lot of similarities between mitosis and meiosis, but there are some very strong differences. Okay, they both begin with diploid parent cells. They both begin with parent cells whose chromosomes have been duplicated, but the end products are different. Mitosis is going to produce 2 genetically identical diploid somatic or body cells. Meiosis is going to produce 4 genetically unique haploid gametes because of that crossing over process that has occurred. Let's compare the two. Okay, here in mitosis, we've got our duplicated chromosomes. The chromosomes aligned at the metaphase plate. Notice that the homologous chromosomes are not lined up together. And then in anaphase and telophase, you're going to produce sister-daughter cells that have the same number of chromosomes the original parent cell had, same as each other. But they're both diploid, 2 pairs of chromosomes. 2 of the long ones, 2 of the short ones here. In meiosis, we're going to have the same replication beginning, okay, but now in prophase 1, we've got this crossing over that can occur here between the blue and the red because of the tetrad that formed at synapsis. Then in metaphase 1, the tetrads align at the metaphase plate. In anaphase and telophase 1, the homologous chromosomes separate, but not the chromatids. The chromatids stay together. Notice that we've had crossing over here, so there's a mixture of the blue and the pink there. And then finally in meiosis 2, after the further second division of the cell, we end up with daughter cells, each of which is haploid. There are 4 of them, and each one is different than the other. They have different combinations of the chromosomes because of the crossing over that occurred back here in prophase 1. This variation in the gametes is one of the reasons that you and all your siblings are not identical to each other, okay? Because you have 2 different things that happen here. You have the independent orientation of the chromosomes at metaphase 1 as well as random fertilization, okay? The independent orientation means that each pair of chromosomes lines up at the cell equator independent of each other. Sometimes the mom chromosome will be facing one end and sometimes the dad chromosome will be facing that end. And it just depends on how they randomly line up. And then when they're pulled apart, then you get a mixture of maternal and paternal chromosomes. You have a lot of different combinations. It says here the number of combinations is 2 to the nth power, where n is equal to the haploid number of chromosomes. For humans, that would be 2 to the 23rd power. You have that many different possibilities of combinations of the chromosomes. Random fertilization is the fact that each sperm cell and each unique egg cell are just, it's just random which ones get together there too, and so you don't have any idea what the combinations are that'll be put together in that case. So here's some probable arrangements in metaphase 1. So here's possibility A and B. You see they're kind of lined up the same way, but in metaphase you might get 2 blue ones and 2 pink ones here, and you might get a blue and a pink there and a pink and a blue in this phase. And then our combinations would be very different, and so you don't really know which combination is going to be which. Another thing that happens, since those homologous chromosomes are separating, that leads to lots of different genetic differences between gametes. Because remember, your homologous chromosomes can have different versions of the same gene. One version could be inherited from the mom, one from the dad. And since they move to opposite poles during anaphase, and that's random the way they line up, then your gametes are going to receive either the maternal version of the gene or the paternal version of the gene. If the parents have the same version, that's great. If they don't, then that's just the difference there between one and the other. So here we have two mice, okay? We have, there are genes for coat color and genes for eye color. This is showing them on the same chromosome here. And so we've got the brown mouse with the black eyes and the white mouse with the pink eyes, okay? And here are the chromosomes on the 4 gametes. And so depending on which direction the genes line up, the chromosomes line up, the offspring could get brown coat with black eyes, white coat with pink eyes, brown coat with pink eyes, or white coat with black eyes, just depending on how it lines up. So let's talk a little bit more about that crossing over thing. Genetic recombination is really important to maintain new combinations of genes, okay? This crossing over is exchanging corresponding segments between separate chromatids, and that's going to cause a lot of different combinations of genes in the offspring. The place where they cross over is called a chiasma. It's not really that important for you to know that, but it's really interesting to see what it looks like in the picture as opposed to the diagram. And what you end up with is basically a breakage of the homologous chromatids and then the rejoining of the non-sister chromatids to each other. And so that's going to end up with some mixing there of those genes that we saw in the mice a while ago. So you can end up with the different combinations there. You got 4 different genetic types of combinations. You've got the brown and the black, the brown coat and the black eye together, the brown coat and the pink eye, the white coat and the black eye, and the white coat and the pink eye there. So you can have 4 different combinations of those genes in the offspring. Some other things that can happen, sometimes there are mistakes that happen, and this is a type of mutation called chromosomal mutations. And so we'll see some examples here of things that can happen with that. When you look at chromosomes, this is called a karyotype. A karyotype is the display of the images of the chromosomes arranged in pairs. Usually they're produced during dividing cells in metaphase, and that lets you look at the homologous chromosome pairs and the chromosome number and structure. And so you can look here at the chromosomes. These are differentially stained to show different things here. And so you can see that they're lined up with the centromeres, and you can see the different parts that are stained differently there that are matched up. This particular karyotype shows the X chromosomes X and Y. So this individual is a male because males have 1 X chromosome and 1 Y, whereas females have 2 X chromosomes. Sometimes there are mistakes in the separation of the chromosomes from each other at meiosis. It's not super common, but it does happen. The most common one is called trisomy 21. This is inheriting 3 copies of chromosome number 21, and it's the most common human chromosome abnormality. It produces Down syndrome. There's a characteristic set of symptoms, including the ones listed here, and the incidence of Down syndrome or trisomy 21 increases as the mother gets older. Here's a graph. There's a graph that will show in your book that shows has the incidence of Down syndrome increases as the mother gets older. In this particular karyotype, we can see here there are 3 copies of chromosome number 21 in this girl, and so she has Down syndrome. This is caused by a process called non-disjunction, and that is the failure of chromosomes or chromatids to separate during meiosis. It can happen in meiosis 1 or meiosis 2, depending on whether 2 sister chromatids go to one pole or both parts of a pair go to one pole. And if you have fertilization after that, then you're going to end up with zygotes that have altered or different numbers of chromosomes. In some cases, this is deadly. In other cases, it causes something like Down syndrome. So here's one type of non-disjunction, okay, when the homologous chromosomes don't separate, and so you'll end up with gametes that have, every gamete have an abnormal number of chromosomes. And then here's the other kind where you have a normal meiosis 1, and then a meiosis 2, you got one of the pairs of chromatids that doesn't separate properly at meiosis 2, and so you end up with about half of the gametes being abnormal and the other half being normal. You can also have the alteration of chromosome structure by just things that break off and don't attach normally. So this can lead to genetic disorders or some kinds of cancer that can result because of this. These can be either a deletion, which is losing a chromosome segment, or a duplication, which is the repeating of a chromosome segment, or an inversion where one part breaks off and reattaches backwards, or a translocation, which is the attachment of part of one chromosome to a different non-homologous chromosome. And this kind of shows you the idea of what can happen. Here's one that's deleted. Here's a duplication where you see 2 copies of that part. Here's the inversion where this part breaks off and is reattached backwards. And then a reciprocal translocation where you get part of one chromosome attached to a different one that are not related to each other.